Thank you all for coming. I'm Emiliano Pereira-Flores. I'm a bioinformatician and a postdoctoral researcher at the Eastern Regional University Center of the University of the Republic in Uruguay. The title of the work I'm presenting here is Anti-Traits Pipeline, Advancing Functional Trait-Based Approaches in Metagenomics. And as the title says, I'm presenting a tool we developed for the computation of functional traits in metagenomic data. This work is a collaboration with other scientists, and these are Albert Barberan, Frank Oliver Glöckner, and Antonio Fernandez-Guerra. This is the outline of the presentation. First, I will introduce what are functional traits, then how can we measure functional traits of microorganisms, then I will present our tool, the anti-traits pipeline, and a brief example application where we use this tool to analyze metagenomic samples of the DataOcean's dataset, and I will conclude with some final remarks. So first, what are functional traits? Functional traits can be defined as measurable properties of an organism that are linked to its performance. And that is, these are organisms' attributes that influence their ecology and evolution and provide information that we can use to study different aspects of organisms' biology. Functional trait-based approaches and theoretical frameworks have been developed to study plants and traditionally applied in plant ecologies. For example, functional traits like leaf size and shape, or plant density, or secondary growth, and others have been used to study the biogeography of plants, or the diversity, or the connection between diversity and ecosystem functioning, etc. And it's even often argued that compared to the taxonomic identity, functional traits provide more information about the ecology of plants. But what is relevant for us here is that all these development to use functional traits to study the ecology of plants provide a theoretical framework that we can borrow to study microorganisms. And to apply this concept to study microorganisms, ideally, what we would have to do is sample a microbial community and measure phenotypic traits, phenotypic functional traits like the cellular size, whether or not they have flagella, or whether or not they're able to sporulate, etc. But as you can imagine, this is technically very difficult. Microorganisms are very difficult to isolate, and these traits are very difficult to measure. So it's clear this is highly impractical approach. That's technically very difficult and impossible. So this takes me to the second part of the presentation, and that is, how can we measure functional traits of microorganisms? And what we can do is compute metagenomic functional traits. These are functional traits that are measured at the genome level of individuals that are randomly sampled from a community. Essentially, this is functional traits at the metagenome level. And there are several highly informative metagenomic traits that can be easily computed. To apply this approach, what we would have to do is sample a microbial community again, but now we have to generate a metagenomic sample. We extract the DNA, sequence it, and obtain our metagenomic sample. A metagenomic sample is composed of short-read sequences for which we don't know the taxonomic origin, and we don't know the genome coordinates, but still this is highly informative data. There are many metagenomic functional traits that can be computed from this data and can be used to characterize microbial communities, or can be used as ecological markers, or to gain insight into the ecology of microbes in general. I listed three here. They're very common: the average genome size, the functional diversity, and the abundance of antibiotic-resistant genes. And in order to promote and facilitate and advance the computation of functional traits, metagenomic functional traits to study microorganisms, we developed the anti-traits pipeline. Currently, the anti-traits pipeline is able to compute 25 metagenomic traits, ranging from GC content and amino acid composition to functional diversity and average genome size. And this work is actually an extension of the previous work of Barberan and co-workers, who assessed how a set of 15 metagenomic traits can be used as ecological markers. This is how the pipeline looks like. There's too much to go through the details, but what I want to stress is that the input is an unassembled metagenomic data. We avoid a metagenomic assembly, which is one of the most intensive computationally tasks in metagenomic analysis. And then the pipeline follows different paths to compute different metagenomic traits. And currently we have 25 metagenomic traits, and all these provide different information about the ecology of microbes. So a possible output would be something like this, where we get a profile of the different metagenomic traits for each metagenome. This is just a figure to illustrate that. And so I was saying that from all these metagenomic functional traits provide information about the ecology of microbes. I will show the information we get from two of these highlighted here, the AGS and ACN. AGS stands for average genome size, and ACN for average copy number of 16S rRNA genes. The average genome size can be used as a proxy to assess whether organisms behave more like specialists or generalists. The specialists are the organisms that have a reduced metabolic repertoire, and the generalists are the organisms that have more diverse metabolic repertoire. They can metabolize many different compounds. Similarly, the ACN, the average copy number of 16S rRNA genes, can be used as a proxy to assess whether organisms behave more like oligotrophs or copitrophs. Oligotrophs are the organisms that grow slow but can exploit efficiently limited resource, and copiotrophs are the organisms that can grow very fast whenever there is an input of a high concentration of nutrients. So I will show you the result we get when we analyze these two metagenomic functional traits in the DataOceans dataset. But first, I have to introduce the DataOceans dataset. And this is one of the largest sequencing efforts currently available. It includes more than 200 metagenomic samples, or marine metagenomic samples, that are obtained all across the globe. And here we analyzed a matching subset of 63 marine prokaryotic metagenomic samples obtained from 21 sampled sites. Well, we have only 21 sampled sites, and for each one of these sampled sites, we have 3 metagenomes. One from the surface, one from the deep chlorophyll maximum, and one from the mesopelagic water layer. So that's how we have 63 metagenomic samples. And these are just different water layers along the water column. So, and what we observe when we analyze the average genome size in these data is that it increases with depth, and also the genome size of different water layers significantly different between water layers. In this plot here, I show the distribution, box plots, of the average genome size in the 3 different water layers. In red, we have the surface. In green, we have the chlorophyll maximum, and in blue, we have the mesopelagic water layer. And the x-axis is this base pairs, the size of the average genome size. Well, we see that the 3 water layers have significantly different average genome size. We have a similar result for the ACN, the average copy number of 16S rRNA genes, but in this case, on the mesopelagic water layers is a greater ACN, significantly greater ACN than the other 2. And considering the information provided by these metagenomic functional traits, what we can say is that, well, these results indicate that in surface waters, we have organisms that have a reduced metabolic repertoire and that are efficient and slow growers. This is known as K strategy in ecology. And in deeper waters, we have more diverse metabolisms and organisms that have a more diverse metabolisms and have the capacity to grow faster whenever there is an input of nutrients. And this is known as the R strategy. So this is actually not new. It has already been observed, but and is in agreement also with the ecological conditions that are known to occur along the water column. But what is relevant for us here is that we show how these very simple metagenomic functional traits that can be easily computed from unassembled short-read data, we characterize microbial communities in this case along the water column, in terms of their ecological strategies. And with this, I reached the final remarks, and these are three. First, that the empty traits pipeline performs systematic computation of a comprehensive set of metagenomic functional traits. These metagenomic functional traits can be used to characterize microcommunities, but also provide information about the ecology of microorganisms. And a tool like this is very useful to monitor microcommunities along time or environmental gradients, or especially whenever there is an environmental perturbation. The empty traits pipeline is programmed in Awk, Bash, and R, and is available in GitHub at Pereira memo/metagenomic pipelines. And many thanks for your attention.